Thank you very much for giving me voice. I would like to talk about microalgae cultivation and how to support using in support and how to extract polymeric substances of this plant. The four microalgae were selected due to the application for production of oil, hydrogen, and phytochemistry production and cultivation. Additionally, how these four mediums can be also after this the primary function use or disuse to produce polymers. The extracellular polymer substances are produced by microorganisms and used in microalgae for bioflocculation. Also, compounds that may be used as carbon substance sources. The study two strategies for harvesting and how is polymer provided and how to if we add such polymers initially from earlier, it will help in bioreactor or how help if we initially not remove but lift this for further cultivation and as a result were observed increasement of content and sedimentation then of recovery and retail uh, extraction of substances for oil or plastic efficiency increased to 72%. The macroalgae, a solution, they can carry and by absorption they can catch carbon dioxide, making reducing greenhouse effect and be as part of leachate treatment in wastewater and sludge management. One of improvement is certification, but is commonly used, so how further we can increase efficiency. But the trouble is that centrifugation also generates much fast, so other techniques which will be cheaper are also used, also desired to make it applicable. The filtration is the separation and membrane fouling in separation and filtration to get is another story because in this extraction lacks to fouling. So these are another challenge in this case. So it is hoped that these polymers can be material for better membranes, longer, more resistance. Both sense of both proteins of extracted could stimulate the aggregation of cell or production of extracellular polymer substances and contributing then for better sedimentation properties of microalgae. Thus the aim was assess possibility of utilization of extracellular polymer substances produced by microalgae and cyanobacteria, which are also bacteria but microalgae source, but more likely to produce the biofuels than hydrogen. The extracted was from activated sludge using an anaerobic reactor with volume of 20 liters, fed with modern municipal wastewater of high organic compounds of 0.7 kilogram curd chemical demands per day, and the oxygen content was 2 milligrams per liter. During operation was shortened time activating sludge 15 to 5 minutes. Then extrapolymer substances were extracted and characterized. Content was by total organic carbon per gram of biomass. So this was measured efficiency. The EPC, the extrapolymer extraction, was made by ion exchange resins. Made the ratio rinsed was to 35 to 0.5 to volatile substance of biomass. The prepared samples were stirred at 750 water per meters for 4 hours in temperature of 4 degrees. Then samples were centrifugated at 10,000 waters per meter and again after 1 minute's break for 10,000 waters per minute for 10 minutes. They were cultivating in these reactors of 2 liters. I forgot to add that the article is application of extracellular polymer substances during cultivation of microalgae biomass. It is written by Professor Marcin Piętębowski from Marian Mazur Olsztyn University. It's published in Applied Sciences. And the temperature in the photobioreactors was kept at 22 plus 1 Celsius for chlorella. The taxonomic biomass changes were controlled and microscope total magnification using alga analyzer. Bioflocculation has to introduce into the photobioreactor of mixed microalgae and cyanobacteria. It was used of a day, while the second variant was after 7 days. So it was 100, then added another 100 after 2 days, and so after all was amounts of extracellular polymer substance was summing in second variant or summing 500 milligrams, while in first variant was only 100 milligrams, and then characterized as phosphorus and chlorophyll. The result of application due to changes of chlorella and 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 algas and chlorella and tetracycline subscorbindamis helped. While in case of cyanobacteria was lower only to 50% by chlorophyll content, while in chlorella and was increased and was reduced by up to 73% chemical oxygen demand. So it helped to remove wastewater in case of microalgae to classify bacteria, but cyanobacteria wasn't too efficient. So this is the some case that in some case it helped very much, while in other not. And how was the adding more 500 to 200, 100? And it resulted that 100 milligram was enough to increase and was better than in case adding to 400 milligrams, but after was similar 100 was similar to 500. So when was added 500 milligrams of extracellular polymers, was the same result after than in case of 100 milligrams of extracellulars initially. So we see that if we add 100, it is not is different two optimums, but 100 milligrams seems to be more efficient. And the and the and the all in all cases the biomass was initially better, just in case of cyanobacteria, the giving more helped in concentration, while both microalgae didn't help. So the microalgae behave different than cyanobacteria. So in cyanobacteria was less reduction of chemical oxygen demand, but adding more was more profitable for growing, for cultivation. But still cultivation of algae, microalgae, so tetrasomes and chlorella was much bigger because it was 4,000 milligrams per liter both, while the cyanobacteria reached only 2,000. So the better was application for microalgae than cyanobacteria. However, protein content was better in case of cyanobacteria. So if we want to get protein, polymeric var, we should add cyanobacteria. And in case we want to obtain galactose, because it was also much better than other cases. But in case of chlorella, we have also as carbohydrates much bigger than other cases. On tetracermis was also better overall concentration of sugar content. But especially chlorella was bigger than tetracermis, and between was cyanobacteria. But if we want to produce characters, we should use these arcoalgas because it's bigger. But if we want characters, then we should use rhamnose, we should use cell bacteria, but in case of mannose, extraction, we should use chlorella. So this is the case, the three cases. So this is with bigger sugar. In some cases, it's better for bulk fermentation of anaerobic. It's other not. So this can be used, which then for hydrogen production to see which sugar is better, content sugar is better for this. So thank you very much for listening to me. So we see that in some cases it helps, in some cases not. Thank you very much for listening.